All right, we got our last little lecture on the cells this week covering the cytoskeleton. And I got three things kind of drawn in here, and this should be a quick one. This is what I studied really, the cytoskeleton, how cells change their shape and move things around inside of them. So this is how cells move and move things around inside of them. And we already saw a little bit of this actually with all of these. So I'm going to go through and label these. What is this over here? This is the actin. This is an actin filament. This here would be keratin. Keratin. Maybe you've heard of that one. And this one would be microtubule. Okay. Another name for this is a microfilament. Microfilament. This would be an intermediate filament. Intermediate filament. Pretty cool. And this was just a microtubule. Doesn't have a second name. All right, this one's a microfilament because it is the smallest in thickness. It's about 7 nanometers wide. 7 nanometers wide. Nanometer is 1,000 times smaller than a micrometer. Micrometer is kind of the cellular scale. Nanometers, we get to the protein scales. This is about 10 nanometers wide. 10 nanometers wide. It's in between, it's intermediate. And then here we've got our microtubules, which are about 30 nanometers wide. They're also, microtubules are hollow tubes. Okay. Actin is made out of 2 strands of monomers that kind of intertwine and interweave between each other. Let me see if I can pull up another picture of this. Yeah, I'm just going to pull this up over here. I've already drawn these out before. Yeah, I'll pull that up. So this is the actin filament, and this is the other one. And together they make an actin filament. These things down here are myosin motors. These are what make up our muscles as well as allow our cells to divide, if you remember that. So that's actin. And actin really, I got some videos here to show you guys. Actin helps shape cells. So here I have this is a video I took of a bunch of cells in a frog embryo. And you can see just how this is all labeling actin in red. And you can see just how kind of pulsatile, this whole tissue is moving. Each one of these cells, green we have the nuclei, red we have the actin. Boom, you see this big tissue-wide pulse. Okay, over here in red we have the membrane labeled. This is the fly embryo, fruit fly actually. And then in green we're going to have this little myosin motor accumulate. And you see it all accumulates in the middle. And this is how the embryo folds in on itself. It's by accumulating a bunch of this little walkie myosin to slide these actin filaments to pull these cells to shrink their top surface, which then causes the tissue to fold in. So that's one way we can cause cell shape change, is through contracting these actin filaments. The other way that I talked about early on was if we have additional monomers being added. So, for example, here and here, down here, whoops, trying to zoom in. You know, if I keep adding on new actin monomers here and here and here and here and here and here, we grow out this actin filament. So we can get growth of the actin filament, which can push. So we can push or pull. Actin can change cell shapes by pushing or pulling on the cell. Pretty cool. And this is actually how a cell crawls. So when a cell is kind of crawling across the ground like this, it's actually pushing out here with its actin and pulling here with its actin. So it'll push out there and it will pull its butt. And of course, it will have a nucleus. This is how cells crawl. Pretty nifty through the actin cytoskeleton. This is how they divide, all that stuff. Okay, keratin. Let's talk about this bad boy. So keratin, you know, I drew it my best here, but it's a bunch of little fibers wrapped around, kind of like a cable of a bridge. Or it's very much like one of these pull and peel candy licorices. It's almost the exact same. It looks just like that. That is what keratin looks like. Some of you may have heard the word keratin before. Keratin, it's right there. That's keratin. Your hair is a bunch of suicidal cells that go all into one little spot, and they die and they stick to each other and they continue to grow out this long keratin filament. That is keratin. It's also your fingernails. That is also keratin. Very strong. And what does it allow us to do? So keratin really strengthens your skin. So if we look at skin cells, let me draw like a little layer of skin cells here. We got one cell. Let's draw 3. 2 cells. 3 cells. We'll draw now another little layer. 1, 2, 3. And then up here they start to get kind of flat. So we won't make them like this anymore. We'll make them like this. And we'll draw one more layer up at the top. These boys real kind of flaky, real kind of flat. And then we got them like even like straight up just like flaking off. Your skin is a conveyor belt of cell death. Your skin is a conveyor belt of cell death. So as you get closer to the edge of your skin, one step closer to the edge, I'm about to keratinize of your skin, the cells they're more keratinized. They have more keratin. Okay. It's pretty nifty. All right, so let's draw in our little nuclei and all these little guys. Nucleus, 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 nucleus. Oh, that one's tiny. Nucleus, 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 nucleus. We won't draw nuclei unless that top layer. All right, so now what is keratin doing here? All right, so let's draw in our keratin filaments. Actually, we're going to draw these little specialized junctions called desmosomes. So these desmosomes connect and link all of these skin cells together to really make your skin strong. Without these, your skin would just fall off. So we got these desmosomes, like so. It's really cool. And then you could probably guess these desmosomes, I'm going to draw it in light gray. I'll draw it in light gray. Are all connected to each other many times over through these keratin filaments. So these keratin filaments, what's going on? I want that. Juice, juice, juice. There'd be more over here. I just didn't draw these. like so. And this really kind of helps spread out the force tied into these cells. Oops, I need some of my desmosomes over here. Go back to drawing. You know, because as you pull on one of these cells, it's pulling on all of these different keratin filaments, which are pulling on all of these different desmosomes to help spread out and diffuse the force. It also really allows your cells to not be subjected to shear force and being pulled apart by like the side, being sloughed off that way. These would have even more keratin. They also start to form keratin granules. These two would be connected to each other. I just didn't draw on those desmosomes. Sorry about that. But you get the idea here. So we got lots and lots of keratin filaments in your skin connected to desmosome junctions. Makes your skin really, really strong. I mean, if you think about it, like these are just made out of, these are just cells on the outside of your skin, and you can't pull them off. Why can't you just, I mean, that's just a little tiny cell. Why can't you just pull that right off? I mean, you could, but you choose not to because that would really, really hurt. So something you can think about this is, it's very similar to like what we do with cement. So cement, concrete, it's very good at compressive force. So, you know, I could compress this paper and nothing will happen, right? It's very strong against that. But if I go in here, I don't want to actually rip that. But if I go in here, like this paper, I can't really do anything to it by compressing it. But if I go in here and I rip it, really easy. A different kind of force, right? The direction of force matters. Like cells can take compression, but they can't really take shear force or tearing force very well. And neither can cement. Cement can take compression, but it can't take tensile force very well. If you kind of take a thin piece of cement and suspend it between two things, it'll like bend in and fold and break really easily. But you can compress it for days. So what keratin allows us to do, just like rebar or steel inside of cement, is it keeps that cement from being able to be like bent by shear force, by tensile force. Sorry. So you can think of keratin filaments as like rebar in your skin. They help support those cells and make them more resistant to forces, just like rebar or steel does inside of cement. Very similar concept. These also function, since there's so many strands wound together, they function a lot like the steel strands of a suspension bridge, like the Golden Gate Bridge, for example. You have many, 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 many thin metal wires bound together to hold up a giant, heavy steel and cement bridge. Very cool. And then just like a little picture of what these actually look like. I showed you actin filaments. Let me show you some keratin filaments. Big, huge, thick fibers all throughout the cell to help strengthen it, to spread that force around. So if I pull right here, it's like I'm pulling on that, and that, you know, and all these other cells. And they help spread out and diffuse that force. Very, very cool. And then, you know, keratin it's made out of a fiber, it's made out of a protein. And proteins are biological organic molecule, right? They're made out of amino acids. And it's funny because proteins actually need lipids. They need to be hydrated, moisturized. So this is a little pro tip. So not only are they strong, but they're also kind of flexible and supple. And here we have a hair filament, which is made out of just a bunch of keratinized cells. So this is actually one cell you can see here that's very keratinized. You can see how this hair filament, which is made out of keratin filaments, and it's just layers and layers of those cellular keratin filaments all stuck together. It's very strong. I mean, this is essentially just protein. And like, yeah, you can pull out a hair and then you can break it, but think about that. That's like your giant hands and muscle forces, and it takes quite a bit to break that protein, right? That's how strong it is. Like, do that experiment. It's pretty freaking cool. Like, it takes a lot. And so it's super strong, but it's also super bendable. And it's elastic. And that's because it also has oils associated with it. Whereas if you're I blow my hair, dry my hair every day. If you blow dry way too much, you're going to dry it out. If you use way too much shampoo and you're tearing off all the protein, biotin and things like that, collagen associated with it, then you're really kind of like drying out that hair and making it brittle and weakening it. So you can think of that like your own skin and your hair health. You want to keep it healthy. You don't want to over soap it. You don't want to be rough to it. You don't want to use a blow dryer too much. And if you do, you want to make sure you're conditioning and not putting too much shampoo in your hair. You don't need that much. You just want just a little beauty tips there for you. I'm a biologist and I was raised by lesbians, so I got to do my little thing. But this is you just look at the difference in these. These split ends, dried out split ends versus these supple keratin filaments. Okay, now let's move on to microtubules. And, I talked about these guys. These guys are really, really important for cellular transport. Really important for cellular transport. And we talked about these little guys. So in one direction, not the band, microtubules do know that they're beautiful. So in one direction, we got a little motor protein. God, that looks like trash. These will walk in one direction. These are kinesins. Kinesin. And then in the other direction, we got this boy. We'll draw him in pink. And this, let me tell you, man, this boy got some big O cheeks now. And I ain't kidding. And then it's got like real goofy legs that come down. Kind of looks like a chicken. Oops. Kind of looks like a chicken. And I'll show you a video of this guy in a minute. He's got big booty cheeks and big chicken feet. Then they walk in another direction. These are called dynines. Dynine. Something like that. Dynine with a Y. Okay. Dynine. They walk in another direction. So these are like really important for cellular transport. The other thing that they're really important for is cell division, separating our DNA during cell division. Separating our DNA during cell division. So just a little brief overview here. We've got, oops, and then I'll just show you some cute, cool videos. This won't take long, I promise. We're almost done. So we got our cell that's dividing. We've got actually these little things called microtubule organization centers. They look something like this, called centrosomes. These are actually just like little specialized microtubules. And then from these, we got microtubules that come out. And these connect to our DNA during cell division. And our DNA, our chromosomes are connected like so. And these microtubules help pull this DNA to each side. So then we can have two cells 1, 2, and each will have a nucleus that reforms. Oops. A nucleus, and they'll have these 3 copies of their DNA. And we'll get into all this stuff at the end, like so. And with kind of like little sad faces. But microtubules are very important for this process. So this is a microtubule. Microtubule. Separate DNA during cell division. Separate, yes, separate. These are specialized short microtubules that other microtubules grow out of. Microtubules. Specialized. These are called centrosomes. Let me draw that boy like so. These are called centrosomes. Things like sperm tails also. We got our little spermo here. We draw a little spermo boy. And then they got this big old long tail. It's called a flagellum. Similar to a bacterial flagellum, but a little different. This flagellum, so we got our sperm. Shit. We got a sperm, and we got a flagellum. This flagellum also full of microtubules. And the way that the sperm moves its flagellum is by having microtubules basically bend a little bit like this. And they're bent by like having dyneins walk kind of along them. All right, so microtubules, very cool stuff. Let me just pull up some pictures and we're gonna watch some little videos here together. So here we go, we got a little microtubule. They can grow, they can shrink. And here we've got some microtubules just in vitro, in glass, not inside of a cell. We take some centrosomes and mix in some tubulin, and we can watch these
microtubules continually grow from the centrosome added to a cell extract.
We are.
Quite suddenly, however, some microtubules stop growing and then shrink back rapidly, a behavior called dynamic instability.
I met the guy who discovered that. Timothy Mitchison, that's him. He came and visited our lab for a while. He said we were the Harvard of the Midwest, talking about the University of Michigan. I thought that was pretty cool. Okay. So now that
is a protein
there we were
that
looking at
binds
how
to the
microtubules grow and shrink back. Very cool. Here we're looking at just the tips of the growing microtubules. So microtubules are constantly growing and shrinking. Everything inside of a cell is way more dynamic than you think. It's all changing. Proteins are getting added and removed all the time. And now we're just labeling just the growing tips of microtubules, and they look like little comets. I mean, look at this. It's wild. Whoops. Play. Looks like fireworks.
EB1 is a protein that binds to the GTP tubulin cap at the growing ends of microtubules. Cells expressing a GFP-EB1 fusion protein reveal the spectacular dynamics of the microtubule cytoskeleton. Note that many, but not all, microtubules in this cell grow from the centrosome.
So that's like the tip of a cell there.
Only the ends of growing microtubules are visible in this experiment. Those that are static or shrinking have lost their GTP tubulin caps and do not bind EB1. In contrast, when all microtubules are labeled with GFP tubulin, the true extent of the microtubule cytoskeleton emerges.
So cool. Looks like a little ghost. You're full of little ghosts.
Both growing and shrinking microtubules can be observed.
Very nifty stuff. Okay, what do I want to watch next here? All right, we'll watch this too. We'll probably watch this again later on, but this is worth it. So this is talking about cell division.
This visualization,
and just the wild machinery involved here. And then we'll watch this real quick video on dynein, and that'll be it for the cytoskeleton.
This visualization isolates a single chromosome at mitosis. The chromosome contains 2 identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle, used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with the directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system.
Time to separate that sausage.
When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
It's amazing that any of us are here and alive. What the hell is going on, dude? This is what we're made out of? Okay, one last thing. This guy. We're gonna just, watch a little video of a dynein walking kind of in close up. Dynein motors walk a little funny. They walk like a drunken sailor. Let's look at this thing. When I was in your shoes, we didn't have these animations of dynein and how it walked, and I had to learn this stuff by drawing it out, and it just made no sense to me. That's how it walks. It's got these big butt cheeks and these flappy little wings, and it walks like a freaking drunken sailor, bro. Just stumbling down the road. I'm a dynein motor with my big booty cheeks. A dynein motor with my big booty cheeks. Shave your belly with a rusty razor. You know what I'm saying? That's what we should do with the drunken sailor. Look, I think that's all I wanted to show you guys. I didn't have a time to talk about I didn't talk about digestion. It's okay. I've already gone too long. Real quick, just a little bonus. There's something called a lysosome. And this is like the stomach. This is like the stomach. Stomach of a cell. It's got a high pH, just like our stomach. Or sorry, a low pH actually. It's very acidic. It's got an acidic pH to digest stuff. So we eat. This is how the cell eats and reuses stuff. Okay. Excuse me, I just burped. It's perfect. Perfect ending. Perfect ending. That was a lot. I hope you enjoyed that. I hope you learned something. We went over everything from like, you know, how cells are made out of macromolecules to prokaryotes, all the way through like how a cell knows how to crawl through a frickin' blood cell, you know, an endothelial cell, and go eat a bacteria and engulf it and then digest it. Pretty wild stuff. I hope you got something out of that. Pretty cool. Okay. I will see you guys in lab. Have a good rest of your week. I'll see you in the discussion posts and in lab. Sayonara.